All right. Good day, everyone, and welcome to our video report. For today, we shall be discussing about um, microorganism in waste management treatment and bioremediation, along with Ms. Nicole Maborang and Ms. Vanessa Tianyo. Now, the content of this video report, beginning with the introduction, which includes the basic history of bioremediation, and the aim and objectives of wastewater remediation using bioremediation, microbes for bioremediation, types of bioremediation, the processes involved in wastewater remediation using bioremediation, the stages involved in wastewater treatment, addition of microorganism, further treatment after bioremediation of wastewater, and the recommendation and conclusion. Now, a bioremediation is simply described as the most effective management tool to manage the polluted water and recover contaminated wastewater using microbes as a helpful agent to remediate the contaminated environment. Now, bioremediation in history, it's a technology using microorganisms, was reportedly invented by George M. Robinson. He was the assistant country petroleum engineer for Santa Maria, California in the 1960s. He spent his spare time experimenting with dirty jars and various mixes of microbes. Bioremediation can prove less expensive than other technologies that are used for cleaning up of hazardous waste. Now, in order for microorganisms to bioremediate, the right temperature, nutrients, and amount of oxygen must be present in the groundwater. The right combinations of helpful microbes can eat the pollutants until it really disappears. Now, bioremediation works on the variety of organic and inorganic compounds. The use of microalgae, for example, in removal of nutrients from different wastes has been described by a number of many researchers and authors already. Now, the availability and supply of clean water is a major concern, of course, throughout the whole world, and is a challenge for most developing countries as a result of high cost of setting up and maintaining standard water treatment for plants. Now, syempre, because there are a lot of factories that are built near a water system, katulad sa dagat, malapit yung mga pabrika, ganun doon, nagkakaroon tayo ng water pollution. And of course, if we cannot deny the fact that when water is polluted, it is not advisable for us to benefit from the water that is being polluted kasi nga polluted na yun. Our health is at risk. So, what researchers have found out is using wastewater treatment, using microorganisms, and bioremediation. So, this is already the birth of bioremediation. Now, we simply have to understand the aim and objectives of wastewater management using bioremediation. So, of course, bakit ba kailangan natin ng water management using bioremediation? And of course, these aims and objectives are really helpful for us to understand why the process exists and how it benefits us. First is that one of the aim and objectives of bioremediation is to affirm the effectiveness of microorganisms for remediation of wastewater. Secondly, the reduction of organic content of wastewater, example, the reduction of biochemical oxygen demand. Next is, of course, removal or reduction of trace organic compounds that are recalcitrant to biodegradation and may be toxic or carcinogenic. So, as I have said, it might be very dangerous to our health. And then, of course, the removal or inactivation of pathogenic microorganisms and parasites. Now, allow me to welcome Ms. Nicole Maborang to introduce to us the microbes that are used for bioremediation.
The organism I will highlight today are all aquatic, so they must be submerged in water in order to thrive. They reside in ponds, swamps, rivers, and oceans, but also in places that we don't think as bodies of water, like places that are simply wet. Of course, we humans are also dependent on water, in a sense that our cells are aquatic, so we carry our river inside of us. And to feel our internal aquatic environment, we drink. And you know your local pond could be the wastewater that is nonetheless a promising habitat for microbes. So that's part of what makes wastewater dangerous for both of us and the environment. Diseases born out of bacteria, viruses, and parasites that can thrive in wastewater, which may also be filled with pollutants like heavy metals or ingredients of our own pharmaceutical and toiletries. So bodies of water have to deal with pollutants whether they be the product of local shifts in nature, death and decay, or just plain feces. So that's why we need these microbes for water treatment and safe drinking. The first microbe is the aerobic bacteria. These microbes have often been reported to degrade pesticides and hydrocarbons, both alkanes and polyaromic compounds. Many of these bacteria use the contaminant as the sole source of carbon and energy. Examples of aerobic bacteria recognized for their degradative abilities are Pseudomonas, Alcaligenes, Sphingomonas, Rhodococcus, and Myobacterium. The next microbe is the anaerobic bacteria. These microbes are not frequently used as aerobic bacteria. There is an increasing interest in anaerobic bacteria used for bioremediation of PCBs or the polychlorinated biphenyls. In river sediments, the chlorination of the solvent trichloroethylene and chloroform. So by the way, PCBs are carcinogenic for both human and marine animals. They are waxy oil found in industrial products. The next microbe is the ligninolytic fungi. So these have the ability to degrade an extremely diverse range of toxic environmental pollutants. So the common substrate used for this microbe includes straw, sawdust, or corn cobs. The next microbe is the methylotrophs. This is a microbe are aerobic bacteria that grow using methane for carbon and energy. So the initial enzyme in the pathway for aerobic degradation, methane monooxygenase, has a broad substrate range and is active against a wide range of compounds, including the chlorinated aliphatic trichloroethylene and 1,2-dichloroethane. And the next microbe is the Deinococcus radiodurans bacteria. So these microbe have genetically modified to digest solvents and heavy metals in the water, as well as toluene and ionic mercury from highly radioactive nuclear waste. Next is the Geobacter sulfuredescens bacteria. This microbe can turn uranium dissolved in groundwater into non-soluble collectible form. Lastly, the the Halococcoides ethingene bacteria. These microbes are used to clean up chlorinated solvent that has been linked to cancer. The bacteria are naturally found in both soil and water and are able to digest the solvent much faster than rising traditional cleanup methods.
Hello everyone, we will move on to the types of bioremediation. We have two, the in situ and the ex situ. From the word itself, when we say in situ, it involves treating the contaminated material at the site. So while ex situ, this requires excavation of contaminated soil or pumping of groundwater to facilitate microbial degradation. So when we say in situ, dun na sa mismong site, kung saan nasaan yung contaminated material, dun na siya mismo ititreat. When we say ex situ, kailangan muna nating i-excavate or meron pa tayong gagawin sa lupa bago natin ititreat yung groundwater. So, we can conclude that there are more disadvantages than advantages ang ex situ because maraming madidisturb na ecosystem, madidisturb ang ecosystem dahil sa kanila. It can cause landslide, it can cause the loss of habitats of the animals, and the like. So this on the picture, you can see the types of bioremediation, the in situ and the ex situ. So makikita nyo sa ex situ, marami pa siyang mga process na ginagawa sa top ng soil. And then doon naman sa in situ, meron lang silang nilalagay na mga equipments, tubo na doon na nila nilalagay yung paipantitreat, yung mga microorganisms. So we have two types of in situ bioremediation. We have the intrinsic bioremediation and the engineered in situ bioremediation. When we say intrinsic, kapag intrinsic, sila mismo yung gagamitin natin. Yung mga naturally occurring microbial populations, yun na yung if-feed natin sila, and then sila na yung mag-i-increase yung kanilang metabolic activity and sila na yung magiging treatment natin dun sa contaminated water. And kapag engineered in situ bioremediation naman, meron tayong i-introduce na certain microorganism to the site of contamination. So kapag intrinsic, sila na mismo, yung mga microbial population na yun mismo yung gagamitin natin. Kapag engineered in situ bioremediation, meron tayong ini-introduce na bagong microorganism. So when we will move on to the stages involved in wastewater treatment. We have the primary, secondary, and the tertiary treatment. When we say primary treatment, this involves storing the wastewater in settling tanks where we add salts are added to encourage solids to cling together through a process called flocculation. So, dun naman sa pangalawa, sa secondary treatment, this residual effluent is pumped to the aeration tanks where air is constantly injected and microorganisms are added to break down the remaining pollutants. So, in the tertiary treatment, this treatment removes the nitrogen by denitrification, processing, and phosphates, usually by chemical precipitation from the effluent. So, dito sa picture na to, mas makikita natin yung stages ng wastewater treatment. So when we say primary, dito natin tinatanggal yung mga plastics, the rugs, the bottles, yung mga solid niya na nakahalo dun sa tubig. And then, dun, yung arrow, tignan natin, nagmo-move siya papunta sa aeration tanks. It enters now the secondary treatment. Dito sa aeration tank, dito mangyayari yung 90% na natitreat na ang tubig because yung grease, yung mga other contaminations are removed through that aeration tanks. And so, kapag nag-move na siya, papunta na siya sa tertiary, doon na nare-remove yung mga nitrogen, yung mga phosphorus, meron na tayong ina-add na mga elements, just like the chlorine contact tanks, like that. And so, sa baba niya, doon yung mga waste, waste yung mga sobrang hindi na kailangan. Another one, may I add, and the secondary treatment. Dito na din kasi natin i-introduce yung mga microbes to uh, to kill or destroy, to break down yung mga hindi na kailangan. Doon nagbibigay ng pollution sa tubig. So, the microbial population for bioremediation. So, ano nga ba tong mga to? So, we are taking advantages of the adaptability to grow ng mga microbes or the flexibility ng mga microbes. Pwede silang mabuhay sa sub-zero temperature, sa extreme heat, sa desert condition, in water, excess oxygen, in anaerobic conditions, basta merong presence ng hazardous compounds. So, yun yung tinitake advantage natin na yung kakayahan na yun ng mga microbes, yun yung ginagamit din natin to break them down to. So, the main requirements are the energy source and the carbon source. So, because of the adaptability of the microbes and other biological systems, these are used to degrade or remediate environmental hazards. So, natural organisms, either indigenous or extraneous, are prime agents used for bioremediation. So, addition of microorganisms, paano ba to? The effluent passes through secondary treatment, settling tanks, gaya ng sabi ko kanina, and then may na-inject na oxidized air. So, this process involves any remaining suspended dissolved solids, including fecal matter. Secondary settling following this process removes any remaining particulates. This process also reduces effluent's biological oxygen demand. The following conditions are needed for the optimal efficiency of the microorganisms and their proliferating sa system. So, ito yung mga kailangan natin sa pag-add ng microbes: a constant temperature, dissolved oxygen content, pH, and then the nutrient level. So, kailangan nating i-check yung mga yon. So, kapag natapos na itong 3 stages na to, hindi daw nagtatapos. Kapag natapos to, it is suitable for irrigation process, for the agriculture, like that. And kapag gusto natin na ma-drink yung water na yon, kailangan muna natin pumasa sa standard ng World Health Organization. Kailangan muna siyang ma-disinfect, ma-filtrate, ma-reverse osmosis, and the irradiation. So, you can read those. So, recommendation and conclusion. It is vital to develop more understanding of microbial communities and their response to the natural environment and pollutants, expanding the knowledge of genetics of the microbes to increase capabilities to degrade pollutants. Conducting field studies of new bioremediation techniques which are cost-effective, and dedicating sites which are set aside for long-term research purpose. So these opportunities offer potential for significant advances. So thank you for listening. God bless.